Hello and welcome to SPARQ Biology. This is one of a series of lessons on biology topics for Year 11s and 12s. Today's lesson is on stem cells. My name is Jessica Malcolm and I am a teacher at SPARQ Ed. SPARQ Ed is a joint project between the Department of Education and the University of Queensland. Our classroom and laboratory are located on Level 2 of the Translational Research Institute in Woolloongabba in Brisbane. Our aim at SPARQ Ed is to engage and inspire students in STEM generally, with a particular focus on biomedical research. Usually, we offer single-day workshops for excursions. Hopefully, students will have the opportunity to do hands-on experiments with us again soon. This lesson is on stem cells. The goal of this lesson is to recognise that stem cells differ from other cells and that they can differentiate into specialised cells to form tissues and organs. These are the specific things you should be able to do by the end of the lesson. Recall the organisation of multicellular organisms, identify properties of stem cells, describe 3 types of stem cells, and evaluate and discuss the use of different types of stem cells. You might have seen this slide at the start of the lesson while you were waiting. If you haven't already, spend a moment revising the organisation of multicellular organisms such as humans. See if you can match these numbers with the letters on the right. How did you go? Now, subatomic particles such as protons, neutrons, and electrons make up atoms, which make up molecules. Molecules make up larger molecules, which we call macromolecules. That's E. So number 1 is E. Number 2 is an organelle, such as a mitochondria. Number 3 is a cell. Cells come together to make tissues. So number 4 is tissue. And finally, number 5 is an organ. Tissues come together to make organs. Then organs come together to make organ systems, such as the stomach being part of the digestive system. And then organs come together to make up the whole organism. I had this slide in here in case I needed to go back to this and refer to it in response to your questions. But it is a timely reminder to let you know that if you haven't learned the function of these organelles, make sure you do. You also need to be able to recognise them in diagrams and electron microscope images. What you need to do here is copy down these 5 important words that you'll need to understand that relate to stem cells. Then spend some time working out which word matches to which definition on the right. How did you go? Let's check your answers. Potential means having or showing the capacity to develop into something in the future. Differentiate is to make something different. An embryo is a multicellular organism just after fertilization, but before it has begun to form body structures such as tissues and organs. Specialize means to engage in a particular activity or serve a particular function. And transcribe is something a cell does to copy the DNA code of a gene into mRNA. The mRNA is used as a template for the cell to make a protein. So transcription or transcribing is when a gene is switched on in a cell. When reading about stem cells, you might see these words used in different forms. For example, you'll see potent used as a suffix in the words multipotent and pluripotent. Cells can be differentiated or undifferentiated. An embryo is described as embryonic. Cells may be specialized or unspecialized. And when a gene is transcribed, we call the process transcription. Everyone starts out as a single microscopic cell called a zygote. A zygote is a fertilized egg. How do we then become such a complex multicellular organism? So let's talk through the diagram. Firstly, what's not shown in the diagram is that the zygote divides and produces more and more identical cells. And that process of cell division is called mitosis. One cell becomes two, two cells become four, four cells become eight, etc. Then the group of cells forms a sphere called a blastocyst. The cells in the inner cell mass, shown here in purple, are embryonic stem cells. They are unspecialized, undifferentiated, and highly potent. We'll talk more about them later. Eventually, the cells move into different positions, forming three layers: the ectoderm, mesoderm, and endoderm. This is the first step in the pathway of becoming what we call specialized differentiated cells. The ectoderm goes down the path of becoming cells of the skin and nervous system. The endoderm forms tubey organs like the lining of digestive and respiratory systems and bladder, for example. Finally, the mesoderm forms connective tissue, muscle, bone, and blood. Cells that carry out specific functions in the body are said to be specialized. Because they are specialized to do a particular function, they must contain the appropriate tools, for example, organelles and proteins, to do the job. You can think about this a bit like a student's academic career. When you're in year 7 at the start of high school, you're all doing the same subjects. At this stage, you are unspecialized, like an undifferentiated cell. But as you move through the year levels, you take steps towards being more and more specialized. By the time you're in year 11, you really start to specialize a bit more by choosing specific subjects. And in those subjects, you need particular tools, which might be a paintbrush in art or safety boots in manual work. Take a moment to look at one of the cell types in the picture and suggest a cell structure or organelle that it might have a lot of in order to do its job. An example of a specialized cell here is columnar epithelial cells, these ones here. They are cells that line the digestive tract and secrete digestive enzymes into, for example, the small intestine. They would have a fair amount of the Golgi body, also known as the Golgi apparatus. This is the membranous organelle that helps secrete proteins out of cells. Nerve cells would have a lot of cell membrane. This is so that they can communicate with other cells. And muscle cells would have a lot of mitochondria because they need a lot of energy. They also transcribe a lot of the myoglobin gene. The myoglobin protein will help the cells grab onto oxygen from the blood. The cells need oxygen to do cellular respiration in their mitochondria. Red blood cells are another example of a specialized cell type. They're also called erythrocytes. Young blood cells have a nucleus, But as they mature into red blood cells, they lose the nucleus after developing hemoglobin, a protein that contains iron and carries oxygen around the body. They need as much space as possible to fit in the hemoglobin and carry the oxygen around. They usually last around 120 days before needing to be replaced. Look at the red words here, young and mature. What key words from today's lessons could you replace these with? You could say undifferentiated blood cells have a nucleus, but as they differentiate into red blood cells, they lose the nucleus. Here's the really important part of the lesson. What are stem cells? Stem cells are undifferentiated, unspecialized cells that have the potential to become specialized cells. They have two important properties. The first is potency. They have the potential to differentiate into a number of specialized cells. The second is self-renewal. They have the ability to go through several cell cycles without differentiating in order to replenish themselves. I mentioned embryonic stem cells earlier. These are contained in the blastocyst. They are represented by the blue cells in the image. Embryonic stem cells are what we call pluripotent. This means they are undifferentiated and have the potential to become any type of cell. These microscope images show a blastocyst on the left and a gastrula on the right. You don't need to know the details about these things, but you do need to recognize what happens to embryonic stem cells. So in the blastocyst on the left, you can see the inner cell mass. That's where the embryonic stem cells are located. They move into 3 different locations within the sphere. Based on these locations, different genes are switched on and off. So we get differential transcription of genes. So the cells are now starting to differentiate, becoming either the ectoderm, endoderm, or mesoderm. The diagram on the left you've seen before. The zygote is originally totipotent because it can differentiate into either embryo or into placenta and umbilical cord. Embryonic stem cells are pluripotent. They can differentiate into any other type of cell. As cells start to move towards specialization, we say they are multipotent, but they are still not fully differentiated. In the diagram on the right, the multipotent and unipotent cells displayed are those of the mesodermal layer only. Mesodermal cells differentiate into either hematopoietic or mesenchymal stem cells. You don't need to know those words, but for your information, hematopoietic stem cells become various blood cell types, and mesenchymal stem cells can become bone, cartilage, muscle, tendons, or fat cells. From the previous slides, we can see that there are 2 types of stem cells: embryonic stem cells and adult stem cells. Adult stem cells have been found in bone marrow, blood, the cornea and retina of the eye, intestines, liver, muscles, nervous system, including the brain, and pancreas and skin. These multipotent stem cells are partially differentiated and therefore less flexible than embryonic stem cells. Their job is to replenish cells only within the tissue in which they reside. Here's a question for you to try. Choose the best answer out of the 4 options. How did you go? The best answer is D. A stem cell is any kind of cell that is undifferentiated and can divide to make new cells. Now let's write down the two main properties of stem cells. The answers are potency and self-renewal. And finally, identify two types of stem cells. So there are embryonic stem cells and adult stem cells. Adult stem cells are also sometimes called tissue-specific stem cells. You might also see them referred to as somatic stem cells. Somatic cells are normal specialized body cells, so using somatic stem cells might be a little bit confusing. Stem cells are important in our body to replace damaged and dead cells. Therefore, can we harness their property of potency to treat medical conditions? For example, we might be able to treat blindness, spinal cord injuries, diabetes, and many more. This concept is called regenerative medicine. This is where we use stem cells to grow a particular tissue or organ in the lab and then use that tissue or organ in a transplant for a patient. Read through the example on the right. This is an article from New Scientist from 2 years ago. Which type of stem cells did they use to regenerate the people's eyesight? That's it, embryonic stem cells. Feel free to pause this here to read some details. You can see here that they used embryonic stem cells to grow up replacement tissue to give the patients and restore their eyesight. They also mention a similar trial where they didn't use embryonic stem cells. In this other trial, they used induced pluripotent stem cells, which is where a normal adult cell that has been transformed or induced to become a stem cell is then grown into the desired tissue. So this woman's skin cells were turned into stem cells, and then the stem cells became retinal epithelial cells. It would appear it wasn't as successful as the trial with the embryonic stem cells, but her disease didn't get any worse. It also says down here that these induced pluripotent stem cells are preferable to using embryonic stem cells because it doesn't involve the use of embryos that you have to destroy. But there is a concern that this method may result in cancer-causing mutations. Shortly, we'll look at evaluating the use of the alternative stem cells in regenerative medicine. Before we do that, let's look at another issue with regenerative medicine and the link with cloning. When receiving transplanted tissue grown from an embryo, there is a risk of immune rejection. This is because the embryo has a different genetic makeup than the patient, so the patient's body will recognize the cells as foreign. However, if we could clone the patient by making embryonic stem cells with their own DNA, we could circumvent or overcome this issue. This is called therapeutic cloning and is done by a process called somatic cell nuclear transfer. The diagram on the right shows somatic cell nuclear transfer. This is when a donor egg cell has its nucleus replaced by the nucleus from a body cell from a patient. So this egg is now diploid, like a fertilized egg, but it has the genetic makeup of the patient. We zap it to trick it into thinking it's fertilized, and then harvest the embryonic stem cells from the resulting blastocyst. So we've discussed embryonic stem cells and adult stem cells, but we saw briefly in that New Scientist article that there are also these cells called induced pluripotent stem cells. So they are an alternative to consider when thinking about using stem cells in regenerative medicine. In this activity, your task is to consider the properties of different types of stem cells and ultimately evaluate them for their potential use in regenerative medicine. To do this, you need to think about the advantages and disadvantages of each type. Pause the video here and copy down this table in your notebook. Make sure the last two rows especially have plenty of room. Coming up, I'll show you some statements that you may put into the different sections of the table. Alternatively, you may decide to complete the table on your own, then check the statements later on. Here are some statements that can be placed in the table. Each statement applies to one or more of each of the types of stem cells. There's understandably a lot of research into stem cells being done around the world. Some of this research is happening at the Translational Research Institute where SPARQ is located. There's a bit of a connection to cancer when it comes to stem cells. Some cancer types appear to be due to malfunctions in the adult stem cells that replenish that tissue. There's also an interesting disease called heterotopic ossification, which is being researched. Heterotopic means other place, and ossification means bone formation. So this is a condition in which bone tissue forms outside of the skeleton, usually in the muscle. When this occurs, it's often after an injury, such as a broken leg or a spinal cord injury. So researchers are investigating mesenchymal stem cells to know more about this. Before you finish up today, these are the 4 success criteria we saw at the start of the lesson. Spend a few minutes thinking about how well you can do each of these, and then try doing a bit more research about the ones you're not confident with. Thank you for watching and all the best with your studies. If you'd like to contact us, our email and website are shown on the screen. Stay safe and see you next time.